Okay, we'll get this session underway. My name's Steve Meikle. I'm from the University of Sydney, and this is Molecular Imaging 2 Neuroscience. We have two great speakers and one okay speaker. Just a couple of quick announcements. We're already starting a little behind time, so I won't go through these in detail, but please turn your phones to silent during this session, just as a courtesy to the speakers. I think the other reminders you've already heard before. So I think we'll get underway without further ado. Okay, well, the next speaker needs no introduction, except I guess there's probably some people that have walked in late. So for you, so my name's Steve Meikle. I'm from the University of Sydney. And the title of my talk is Instrumentation for Preclinical and Clinical Neuroimaging. And we debated about whether any of us actually have any disclosures we need to make here. But just in the interest of transparency, I have no financial interest to declare. Our institution is negotiating affiliation and collaboration partnership agreements with MR Solutions, and I have had associations with Siemens in the past, but not currently. So for my talk, I just have a very simple outline. I wanted to just have a little bit of a look at where we are now in terms of molecular imaging instrumentation for the brain. How did we get here, and where are we going to next? So this was a diagram that we published in a PNB topical review more than 10 years ago now. And it shows a kind of a map, a sensitivity kind of resolution map of all the molecular imaging modalities that were available at that time. And just to explain it a little, so what we're looking at here is the sensitivity for detecting in vivo probes in vivo on the y-axis, and what I call spatial scale on the horizontal axis. So at the bottom end, the bounds of these footprints, if you like, represent the limiting spatial resolution for each of those modalities, and the upper bounds represent roughly the kind of sensitivity limits. And one of the things the diagram shows is that for several of our imaging modalities, we're able to span resolution scales that allow us to just about image the mouse and all the way through to performing human imaging. So with some of these modalities, if the radiation or the source of the signal has sufficient length scale in the human body, you can perform clinical imaging. Some techniques aren't quite capable of doing that, some of the optical techniques. But in this talk, I'm going to be having a particular focus on radionuclide-based molecular imaging, since this is a nuclear medicine conference. And I'm going to have a bit of a look at what's happening to sort of push the boundaries of PET and SPECT. So we're all very familiar with these techniques, but just to quickly summarise some of their key features, PET, as we know, has exquisite sensitivity, has really good depth penetration, so we can perform clinical imaging as well as preclinical, and it's intrinsically quantitative. On the downside, it's a very expensive imaging modality. It requires access to a cyclotron and/or a generator. It uses ionising radiation, so we always have to think about the radiation dose to our subjects. It has some factors that limit the spatial resolution, which we'll discuss a little. And it doesn't have the ability to do multiplexing of signals, so we only get one isotope or one radiopharmaceutical at a time, although there have been some attempts to implement some novel methods that overcome that. SPECT has many of the same features, has excellent sensitivity, not as high as PET, but compared with the other molecular imaging modalities, it's still pretty good. It also has good depth penetration. It can do multiplexing, so we can image more than one radio tracer at a time using energy discrimination. It's quantitative and it has clinical utility. On the downside, again, it's somewhat expensive, but not as expensive as PET. Uses ionising radiation, and it has some limits to the spatial resolution that can be achieved. These figures, by the way, come from an excellent review article by Michelle James and Sam Gambier. But I just want to point out there's one point that I actually disagree with them on, and that's this. So in the review article, just to point out a difference here, SPECT was listed as being non-quantitative and it was a con. But in fact, SPECT is intrinsically as quantitative as PET. I think the differences lie more in the traces than the physics or the methods. But that's just a slight difference. So that's SPECT and PET in very, very brief review. And of course, we use them in a range of different applications. This is in very, very general terms. PET is generally very well suited to studying small molecules in vivo because small molecules have fast kinetics. And most of our PET isotopes have short half-lives, so you can nicely match the half-life of your isotope to the kinetics of the molecule. SPECT traditionally has been used more to study larger molecules, macromolecules, antibodies, fragments, partly because in the past they were easy to radiolabel by iodination, and also because SPECT isotopes typically have longer half-lives, so they're again well suited to the biological half-life of the processes that we're interested in study. That paradigm has been mixed up a little just lately, and we're increasingly using PET for studying larger molecules, including antibody fragments, and similarly there are analogues that can be labelled with SPECT isotopes of some of the small molecules in the brain that we're interested in. So where are we right now? So let's just have a look at what we're able to do, at least in terms of spatial resolution, with current state-of-the-art PET systems. So we've got the human brain, rat brain and mouse brain roughly to scale here. And with human PET systems, the current sort of standard state-of-the-art system, we can achieve about 4 to 5 millimetre spatial resolution and get images looking a little like this. And with preclinical PET systems, typically we're getting around 1 to 1.5 millimetres spatial resolution, which gives us nice images of the rat brain. We can resolve the important structures that we're interested in. We don't do quite so well in the mouse brain. If you don't get down to around 1 millimetre spatial resolution, you get images that are quite difficult to interpret in the mouse brain in terms of identifying the important structures. So there's a bit of a boundary there. Looking at the same thing with SPECT, so with human SPECT, current commercial SPECT systems, we can get about 10 to 15 millimetres spatial resolution and get images that look a bit like this. We actually do a little better in the preclinical domain with SPECT. We can get down to spatial resolutions well below 1 millimetre with current state-of-the-art preclinical SPECT systems. And we'll have a little look at a moment as to why that is. And so we can produce really nice images of both the rat brain and the mouse brain, but just keep in mind there is still a very big sensitivity gap there compared with PET. But if you're able to image for long periods of time and or give a bigger radiation dose, you can achieve very good imaging results in both of those species. I also want to just point out that there are other systems coming that are actually on the market but not yet FDA approved. And so there's a system, for example, coming from the people who developed the USPECT system from MI Labs, which claims to have spatial resolution in the human brain getting down below 5 millimetres. So really quite competitive with PET resolution. But at this stage, there aren't any publications of studies in the brain demonstrating that. Of course, we also have the capability to do multimodality imaging, and there's been a huge focus on that over the last 10 years or so. For the brain, PET-MR is particularly interesting and useful. So we now have the availability of commercial PET-MR systems for small animals that allow us to do simultaneous imaging and really make the best use of complementary information from both of these modalities. And we also have clinical PET-MR systems available that are able to do the same in the clinic, and there are many groups studying these, the applications of these systems to just find out exactly where the applications lie. The other advance that I would nominate is the ability to study awake animals. So PET in small animals, when you're interested in the brain, is particularly challenging because typically we use anaesthesia to keep the animal still, but the anaesthetic agents we use interfere with all the sorts of things we actually want to study. They interfere with blood flow, neural hemodynamic coupling, as well as many of the neurotransmitter systems that we're actually trying to study. So there's been quite a few different approaches to try and tackle this problem. This is our own work on using motion tracking techniques to do motion correction of awake animals during PET studies. This is some work from Chiba in Japan where the approach is a little different. The idea here is to keep the head firmly restrained, so the animals have surgery and they have an attachment to the skull that keeps them rigidly fixed to a head restraint device. And so that requires a lot of training and acclimatisation, but studies can be done with the animals in the conscious state. And then, of course, we have the RatCap system, which was developed at Brookhaven. And the approach here was different again, and the idea was to develop highly compact miniaturised PET detector systems and attach the ring of detectors directly to the animal's skull. So this obviously involves invasive surgery and a lot of training and acclimatisation as well. But the idea is that the detector ring is then supported by a counterbalance system, and you can do imaging of awake and, to some extent, freely moving animals. So there's some of the approaches. So this is a little bit of an extension of a method that we've developed, which we call open field PET. And now the animal is in a chamber and it's free to move around in any direction. I'm just going to really quickly show you what it's capable of doing. So you see the animal moving around, and we're tracking a marker attached to the animal's head continuously. And at the same time, this robot is repositioning the chamber in response, in adaptation to the animal's movement in order to keep the brain in the field of view of the PET scanner. And what you see below is, this is a C11 raclopride study. So this is an image of the raclopride study superimposed on an MRI template. And this is the first 20 minutes of the study. So you're seeing specific binding there in the striatum, as you would typically see. So these images have been motion corrected, and they're of a very high quality. This is the image taken during the period after a competing drug has been injected. So this is a drug that competes for D2 binding sites with raclopride, and you see extinguishment of the specific binding signal. These are the time activity curves from 4 animals that underwent this type of procedure after we've normalised the curves for injected dose and weight. And you see how reproducible the time activity curves are. This is the striatal curve, where you see a significant displacement. The drug is just cold raclopride in this instance. And this is the cerebellar curve. So you can see it's a very reproducible technique. And this is the output from the kinetic modelling. I won't try to explain this. Evan, who's going to follow me, is going to do that for me. But these curves, these individual grey curves, show the raclopride displacement from D2 receptors of each of the individual animals, and the red curve is the mean of those curves. So you can see we can extract interesting non-steady state parameters from these types of studies and estimate things like the time of onset of the displacement, the magnitude of this displacement, and the duration. I'll show you an actual research application of this technique a little bit later on. Of course, there's been a lot of work in that space as well in other imaging modalities, including optical and focused ultrasound. So there's a lot of activity in this space, and essentially the aim is to try and combine imaging of transgenic animals with behavioural studies. So it's a really interesting and exciting development that's taking place at the moment. Now, in terms of how do we get here, I'm just going to briefly go through what some of the parameters are we're trying to optimise and how we've solved some of the problems. In SPECT, the go-to geometry is the pinhole collimator. So it's the pinhole collimator is ideally suited for small animal imaging because it enables you to get your target very close to the aperture. And so as you do that, as you move, if you've got a small target and you can move it close to the pinhole aperture, you get a rapid boost in sensitivity as you get closer. And at the same time, the closer you get, the better the spatial resolution gets because of a magnification effect. So you're magnifying the source distribution on the detector. So you can boost both spatial resolution and sensitivity by using pinhole geometry. And there are a variety of approaches to that. So here's a single pinhole. Most commercial systems use multi-pinhole imaging, so you've got more than one pinhole aperture projecting the data onto the detector and making better use of the detector real estate. And some systems allow those projections to overlap and then use image reconstruction techniques to uncouple the overlapping data. And there's a really wide range of performances that can be achieved amongst all the commercial SPECT systems. So this is a graph taken out of an article by Fred Boisson that was published a few years ago that showed the performance in terms of spatial resolution versus sensitivity of a number of commercial systems that were available at that time, really just showing that there's a number of for each system there's a different approach to pinhole imaging, and within each system you can have a variety of pinhole apertures and numbers of pinholes and different geometries, and also detector technologies, and all of these combined give you quite a range. But you can see that it's quite achievable to get below the 1 millimetre spatial resolution limit, and some systems have higher sensitivity than others. Now, returning to PET, the main issues here that we need to consider when it comes to the factors that affect spatial resolution are not just the geometry but also the physics of positron annihilation. So I've exaggerated this effect here, but of course the positron travels a finite distance in tissue before it annihilates. So we've got some uncertainty there in our localization just due to positron annihilation. We also have this angular distribution of the annihilation photon, so there's a slight deviation around 180 degrees that causes some additional spatial uncertainty. And then we have the actual size of the detectors themselves, the width of the detectors. And these factors all combine in quadrature as shown by this equation. To just give you a quick example, so if you were imaging fluorine 18 in an 80 centimetre detector ring system with 4 millimetre detectors, you'd get numbers like this. And I just want you to note the acollinearity effect here is quite substantial. It contributes about 1.8 millimetres to the overall spatial resolution. And if we combine all that in this equation, we get a limiting spatial resolution that's around 2.7 millimetres. In practice, it's significantly worse than that. It's closer to about 3.5 to 4 millimetres in practice. So one thing we can do is to reduce the diameter of the detector ring, and that helps us in a couple of ways, in particular with the acollinearity effect. So when you reduce the detector ring, you can also use thinner crystals if you want to achieve higher spatial resolution. So if we take a similar example that's a little bit more geared towards small animal brain imaging with a 12 centimetre detector ring and typical 1.5 millimetre size detectors, now you see the acollinearity contribution is only 0.26 millimetres. The detector contribution is now about 3/4 of a millimetre, and the overall resolution is below 1 millimetre. So that's largely how the high spatial resolution of preclinical systems has been achieved. And again, in practice, we get something that's a little closer to just over 1 millimetre spatial resolution. And of course, an important part of development and the solutions has been high-resolution detectors. So this slide shows a typical human PET detector from a few years ago with coarsely segmented crystals and a small number of photodetectors sharing the light output. Then we have one of the earliest commercial small animal PET systems with more finely segmented crystals, and in this case using position-sensitive photomultiplier tubes and light guides. And the detector on the right is one that we've been developing in collaboration with Simon Cherry's group at UC Davis to put into our mouse PET system, which uses crystals that are now well below 1 millimetre on the side, and we're using silicon photomultiplier arrays to read out the light from that array. We'll come back to that detector in just a moment. The problem, though, is that if we reduce the ring diameter and make the crystals thinner, we now amplify another effect, which is the depth of interaction effect. And that is that when the annihilation occurs, we look at a line of responses off from the centre of the field of view. We hit these crystals, but we don't actually know where along the length of those crystals the annihilation's endpoints were absorbed. And so we have a number of possible combinations that could explain this coincidence event, and that leads to this radial blurring of the point spread function. So now we've got uncertainty about the positioning, which degrades the resolution as you get further towards the edge of the field of view. So it particularly impacts on ring systems where there's a small ring diameter and you're using long, thin crystals. To overcome that, we use depth encoding detectors, and there's been a lot of work on this over the last 10 years or more. This slide shows some of the approaches to solving this problem, including detectors with dual layers of crystals that are offset in their crystal pitch. Another approach is to use 2 layers of different scintillator materials with different timing properties, different light decay times. Alternatively, we can use the same crystal and coat 1/2 of the crystal block with a phosphor that again manipulates the timing of that half of the crystal block. And the other approach is to use photodetectors that are compact and are able to read out the light from both sides of the crystal array. Another approach is to use monolithic crystals. And here the aim is to estimate the location of the detection in 3 dimensions, and you do that by looking at differences in the light distribution coming from this monolithic scintillator. So there's lots of ways you can solve this particular problem. The detector I referred to earlier on that's going into our system uses the dual-ended approach, so we've got silicon photomultiplier arrays on both sides. So this is the SciPM array. You can see here we're able to identify each of the individual crystals, which are 0.785 millimetres, so we're doing a good job of separating the crystals there. But more importantly, this is the DOI data from this detector. So it's showing the... So the way it works is you're using the ratio of the magnitude of the signals from either side, the 2 SciPM arrays, and that ratio gives you information about the depth of interaction. So these are the depth profiles of this detector at different positions or different depths in the crystal array. And the average DOI resolution for this system is 2.8 millimetres near the centre of that detector. The other thing I mentioned earlier on is the ability to do multimodality imaging, and so when we use these silicon photodetectors, they're highly immune to magnetic fields, and so that's what's primarily given us the capability to do PET-MR systems, whereas the more conventional photomultiplier tube-based detectors are highly sensitive to magnetic fields. So that's where that development has primarily come from. So just in the last 5 minutes or so, I just want to talk briefly about where we're going, what direction is the field going, and I guess whoever gives this talk would have different opinions about this. This was a talk I gave just a little over a week ago, kind of addressing this question. The title of my talk was The Incredible Shrinking and Expanding Pet. The expanding part of it obviously is the Explorer system, which Simon spoke about yesterday in great detail. So I'm not going to repeat what he said about that system, but I think that's a really nice example of, in not only the clinical but also the preclinical space of trying to make the most use of the available signal and pushing the sensitivity of our PET instruments way beyond what we're able to achieve at the moment. And the important thing is the applications that opens up. And similarly, at the other end of the scale, the shrinking part is the move towards developing higher resolution systems for imaging, in particular the mouse brain. And in our case, we're also interested in combining that capability with behavioural assays. So we want to study awake animals. So this is the system we're working on with Simon's group, and I won't try to describe this in any great detail, but one of the unique features is that it will have a touch screen at one end so that the animals can do instrumental conditioning experiments while we're studying brain physiology with PET. Now, just to give you a little bit of a taste for what that might do, so these are studies we've done in the rat brain with our current PET system. And so these animals have been conditioned with, it's a Pavlovian or classical conditioning study where the animals have been conditioned to associate one of three sounds with one of three specific reward outcomes. And then during the PET study, they hear one of those sounds at a specific moment in time, 20 minutes into this PET study, and it's a raclopride PET study. So on the left, and this is the particular study where the animal had been conditioned to expect, with this sound, they expect a reward every time. And so you'll hear that sound in a moment. So this is near the beginning of when the stimulus occurs. So you see the animal get really excited because it knows there's something coming. And then on the right-hand side, this is just about a minute and a half later, right at the end of that 2-minute interval. And it's at the very end of the 2-minute interval that the reward is actually delivered. So you see him, he knows that there's something coming and he's going for the reward, and he knows which side as well. So we're imaging these animals all the time, but we're also measuring their behaviour. So these are graphs showing how often the animal is checking the left and the right receptacle for a reward. And this is the condition where it's expecting a reward, so it's checking often and it's checking the correct receptacle more often than not. It checks less often when it knows that there's only a random chance of a reward being delivered about half the time. And it checks even less often when it knows or expects that there won't be a reward coming. And in these experiments, we mix that all up. So sometimes they're expecting a reward, but they don't get one. Other times they're not expecting a reward and they do get one. But anyway, here's just the data from one raclopride PET study. And all I really want to illustrate here is that it is possible to measure, through kinetic modelling, endogenous dopamine release during this kind of paradigm. So we're seeing a significant displacement of the raclopride caused by dopamine release here, and maybe just a barely significant dopamine release here, and these are 95% confidence intervals, so it may or may not be significant. So I think there's a whole bunch of interesting applications waiting for us there. And now the one parameter I haven't mentioned at all is timing, and there have been other talks talking about this. I didn't touch on it much because in the small animal imaging area, particularly in the brain, time of flight doesn't give us all that much. However, I think it's worth pointing out that the guys who are really pushing this, people like Dennis Schart at TU Delft, have broke through the 100 picosecond timing barrier some time ago now, more than 3 years ago. And that's really interesting because when you get down to below 100 picoseconds, you're now talking about what that equates to in terms of distance is around 15 millimetres. So that starts to become, there's the potential there to use time of flight information on a scale that's relevant to small animal imaging. But the caveat here, of course, is that this is in the lab, it's highly idealised, and it's just a pair of detectors. So the challenge is to translate this to full ring systems and implement it in practice. So just returning to that diagram I showed, so I think we've made a lot of progress, and I guess what the message I wanted to get across is it often feels like all the problems have been solved, but I think we've still got quite a long way to go in terms of pushing the performance of our SPECT and PET systems. For example, PET now is more like here, so we're below the 1 millimetre resolution limit, which is actually really important for mouse brain imaging. And we've seen through projects like the Explorer and other approaches that we're also boosting the sensitivity, which we all thought was already good enough, but there's still a long way that it can be improved. And the important point, I think, is that this opens up a lot of applications in both the preclinical and clinical space. So I just want to acknowledge some people who contributed slides or data to my talk: Bernd Pichler from University of Tübingen, Simon from UC Davis, Dennis Schart, and also the funding agencies that have funded our work: the Australian Research Council, the NCRIS scheme that funds the National Imaging Facility, and the Australian Institute of Nuclear Science and Engineering. Thanks for your attention. All right, I'll put my chair's hat back on. Unfortunately, the last speaker has just gone over time. But we probably have got time for one question. Tian Yu, go to the microphone if you wouldn't mind. Thanks.
Yeah, thank you for excellent education, Dr. Miko. So my question is, how do you forecast the future of SPEC technology? Do you think that will also be shrinking and expanding in the SPEC systems?
Yeah, that's a good question. Yeah, look, I think there's still a lot of potential, and I think in SPECT, I think really the huge gains to be made are in sensitivity. It's still, even with these multi-pinhole systems, I mean, if I think, for example, of the latest USPECT system, it's a really smart design with, I forget how many pinholes now, maybe up to 150 pinholes, highly focused, using every little bit of detector real estate available, and they give you great images, but it's still more than an order of magnitude down on PET for sensitivity. And although you can do imaging with stationary detectors and get great images, you'd really want to boost the sensitivity if you want to do dynamic quantitative studies. And I think the applications will open up if we can bridge that sensitivity gap, or at least keep improving the sensitivity. Spatial resolution, it's always possible with pinhole geometry and smart designs to improve where we are, but I think sensitivity is where it really needs, and there's a lot of potential to improve things further. Okay, in the interest of time, I think we'll move on, and I'll introduce our final speaker, who's going to try and explain all the modelling stuff that I couldn't or didn't have time to explain. So our next speaker is Evan Morris from Yale University.
All right, so this is going to be a little modelling, a little application, less of a grand picture of anything. And the first thing that I now do is I acknowledge my grants and my collaborators on the first slide, because I always forget to do it at the end. And on the left is a list of all the PhD students and a few postdocs that have contributed to this work. So now I've done my duty. And if you didn't download the app for this talk is right there on the right. And that's a little navigation of where I'm going to go. And first, I'm going to do a little tiny bit of review. I'm looking for, there we go. Okay, a brief review. And this Australian ibis was, I was running around after him in the botanical garden in Sydney to get a good picture, and he will come back a number of times during the talk. So first, before we get to the model and what I'm going to say about smoking, we have to do just a tiny bit of review in case anybody has forgotten what we typically measure with receptor ligands in PET, which is something called binding potential. And the theory of binding potential is this, that we're measuring some parameter which is theoretically, at steady state, it represents the ratio of bound to free tracer. And the tracers are those triangles. You know all tracers. This is what I know about chemistry. Tracers are red triangles. And now we interpret binding potential typically as the product of the available number of receptors and the affinity. Okay? And we estimate it as a ratio of two rate constants. Okay, so there's what it is theoretically, how we interpret it, and how we actually measure it. But the real question that the IBIS would ask is, what is this thing good for? And I'll try to explain very quickly. First is we use this index as a measure of receptors. So in those diseases that you know very well, where there's a loss of neurons and a loss of receptors, what we find is that if things are working, binding potential goes down, and that's a measure of loss of receptors. On the other hand, we can also use this same index, oftentimes, as a measure of neurotransmitter release. And what I know about physiology is that neurotransmitters are green triangles. And so when they proliferate, when they're elevated, there's also elevation of neurotransmitter by displacement causes reduction in binding potential. And we can, so the endpoint we actually measure is this fractional change in binding potential from condition 1 to condition 2. And finally, as we heard about before, sometimes we use PET and receptor ligands for drug occupancy studies, and we compare a baseline condition to a drug condition, and drugs, of course, you know from chemistry class, are yellow triangles, and they also displace the tracer, and increase in drug concentration also translates into reduction in binding potential. And occupancy turns out to be the percent change in this fabulously useful but possibly dangerous binding potential index. Okay, and the word to key on in this slide is dynamic. Our signals that we acquire over time are dynamic, and they're made up of multiple components of tracer in different states. And when the signal changes, we can sometimes, through the magic of analysis, attribute that change in the dynamic signal to a change in some physiological parameter. And so the IBIS already has a sense that this has something to do with models. But the end of this review is just to remember that if all works well, binding potential, that typical endpoint of PET studies, can measure receptor density or neurotransmitter change or drug occupancy. But that's not always, it doesn't always work. So let's focus now on cigarette smoking, which is what my colleagues and I have chosen to study with PET. And we have to go back and look just historically for a moment at the studies, at least as of 2010, that had been done on cigarette smoking or nicotine gum or something like cigarette smoking, all using the tracer raclopride, which you heard about before. And there's something very troubling about this table. It's that on the right, basically, if you fell here from Mars and read all the papers on smoking and raclopride, you'd think that the humans don't know anything about smoking and raclopride, because all the results are completely inconsistent. So we asked, why so much variability? And there are little differences between these studies, and they used slightly different models, which we'll talk about in a moment, but essentially they were all measuring change in binding potential, which was worrisome, and there was not any agreement. And in fact, the thing that was most curious, was that the same group had done the study 3 different times, and they themselves didn't get the same answer every time. So that was really curious and piqued our interest. Okay, so at this point I would like you to think, geez, maybe there's something a little bit different about the effect of nicotine on the dopamine response that's unlike the classic challenge that you associate with PET studies, which is amphetamine intended to raise dopamine. All right? And what could it be? Well, so simulations are always the place that engineers start. And this is a simulation of data at 1 voxel in a raclopride study where the smoking happens at 45 minutes. First of all, it's noisy as hell. That's worrisome. But the model, whatever classical model you apply, you can see when it measures a deviation from baseline with just a little bit of the data, there looks like there's some substantial deviation due to the cigarette smoking. But if the model is applied to a larger window of data, you can see that the apparent change from baseline seems to be getting smaller and smaller. And if I take away the noise, it looks like this. And now you're already sensing what I'm getting at here, which is that if you have a dip caused by a stimulus and you apply a model that can't dip, it's like can't do the tango, can't dip, then you're going to be highly dependent on what we called the data window. And this susceptibility, this inadequacy, this weakness in all the kinetic models is consistent across all of them that are using binding potential as an outcome measure. They're all highly sensitive to the data window that you choose if there's something special in your signal which is caused by nicotine and not by amphetamine. All right? And so remember those 3 studies by the same group which had different results. And we looked and we looked and we looked for a long time until the papers that they had written were threadbare. We looked at them and handled them so much until one of my graduate students found this slight difference. In their first study, they only looked at 10 minutes after the stimulus, and they figured, well, if 10 minutes is good, 30 minutes must be 3 times as good. So in their subsequent studies, they looked at 30 minutes after the stimulus, and you could see they appeared to get a reduction in the effect compared to the first study because it was being washed out. All right? And those numbers are consistent with our simulations. So I would say, or the IBIS would say this, it's a good news, bad news story. The bad news is that binding potential, this sort of omnipresent, always used, always reliable index of binding may not be robust as a measure of things that change rapidly. But the good news is it means that there's actually timing information in the PET data. All right? So now we come to the need for a new model that's better tuned to what it is we're trying to measure. Okay, and this model, for historical reasons, we call LPNT-PET, which is an unfortunate mouthful. But it's a model that's designed for those cases where neurotransmitter concentrations are changing rapidly and to be used at the voxel level. Okay? And I'm just going to talk about 2 aspects of the model which I think are most important. One is computational simplicity, and the other is this oftentimes unappreciated need to correct for multiple comparisons. And so when we talk about computational simplicity, we chose to try to describe our data with a linear model. And that's because computationally we could do it at every voxel quickly to make what we thought would be parametric images. And from a historical standpoint, I just point to this, one of the most famous linear models in PET, which you'll all recognize as the Logan plot. It started out as differential equations, but through the magic of integral transforms, it becomes y equals mx plus b, where m, the slope, is an important physiological parameter. That's the beauty of integral transforms, and that's what I mean by a linear model. Now, of course, Gene Logan came after Cliff Patlack, and after Gene Logan, there was Roger Gunn and Masa Kise, and especially for my purposes, Nat Alpert and Mark Normandin, but we don't have time for all of their papers. So I'll just say that if this paper can see far, or even a few feet, it's because we stand on the shoulders of giants. And so this model, which I'm going to show you in a moment, is, as I said, it has a lousy name, LPNT-PET, but the idea is to try to apply it in those circumstances where there's a transient release of dopamine during your PET study. And it looks like this, simply. On the left-hand side is a typical linearized model, which can be solved computationally easily. And the right-hand side is some special term for the dopamine phenomenon. And so you can think of it as a mashup of two models, a model of tracer uptake and another model of dopamine change. And that's what we're going to apply, if it works, to our data, actually during smoking. All right. The other thing is, because we want to do this at the voxel level, there's all kinds of comparisons, and we chose to take the approach of applying using a cluster size threshold to determine what would be significant and what happened just by chance that we'd have to throw out. And so the IBIS is reminding me, I haven't actually shown you what the model produces, except that Steve came before me, but it produces something like this at every voxel. It estimates parameters related to the tracer kinetics, but what we really care about is it also estimates a curve in time, which is the dopamine response, that fast, that transient thing, due to whatever stimulus we're applying. And this curve, is characterized by parameters. There are timing parameters like the takeoff, and there are magnitude parameters like the peak height. Okay. And again, when we say multiple comparisons, what exactly are we talking about? Well, so at every voxel, if this works the way we intend, we're going to ask this question, which is, does the dopamine curve that we estimate improve the fit of the data? And if it does, compared to the models that don't have this flexibility, then we're going to call that dopamine activation. Now, that sounds, I phrase it sort of like a statistical question, which means you need some sort of statistic. And the statistic that we've hit on, that we use, is the F statistic. And it looks like that, but it doesn't matter exactly how you calculate it. The F statistic asks this question arithmetically. It asks, if the dopamine response in a given voxel, the dopamine response in a given voxel is considered significant if the presence of that term, using that term in the model, appreciably lowers the fit, improves the fit, lowers the sum of squares. Okay? So that means we're going to do that F statistic at every voxel. That's where the multiple comparisons comes in. That's where the danger, the possibility of false positives comes in. All right? And so to control false positives, we can take an approach which says that these false positives will cluster next to each other in neighboring voxels. The bigger they cluster, the less likely that could have happened by chance. One lousy voxel can have a false positive. It happens all the time. We throw it out. If we see a response in neighboring voxels, enough of them, such that it happens only p less than 0.05, then we take that to be a real phenomenon. Okay, and another way of saying it is that by choosing our cluster size threshold for this voxel-wise method, we can control that's maybe on the next slide, we can control our false positive rate, and for our study that I'm going to tell you about now, we chose to control it to limit ourselves to no more than one false positive cluster of activity in 10 subjects, which in retrospect I think is too conservative, but be that as it may. Okay, so now you've been here already, I think I've been talking 13 minutes, and you're asking yourself, well, where the heck are the results? So here they are. All right, so this is applied to smokers smoking cigarettes. And okay, so that's the design. People come in, they're abstinent from overnight. They're injected with raclopride. At about 35 minutes into the scan, they start smoking their one cigarette. This is in the scanner. They're smoking their cigarette of choice. So this is self-administration for all the psychologists in the audience. And then the model is essentially asking, is there a dopamine response in that window that's got the little bracket that we can detect in our data? And this is what it actually looks like, because we're doing this in an HRRT, and we're doing high-frequency motion detection so that any movement of the head is not mistaken for cigarette smoking effects. And the people have to do some training so that they're not, we're eliminating the novelty aspect of this. We're venting secondhand smoke so that we don't kill any of our techs. And we're asking some behavioral questions during, before, and after the study. And it looks like this. If you wanted to peek in, that's what it would look like. That's when I had more hair. And of course, the most important part is the graduate student who has the unfortunate job early in his or her career of catching the cigarette ashes. But this is like a trial you go through. It's trial by fire, sort of. Okay, and for those of you who are students in the audience, this could be you. If you're interested in doing this, you can talk to me afterwards. Okay, so let's see, how does this model perform on people? We're going to look just in the reward area of the brain, voxel by voxel, and first we're going to look at the same person who came in twice, once to smoke and once where they just laid in the scanner and didn't do anything. Okay? And because, remember, I have this curve over time, I have 4D data, which I can now display to you as a movie, right? If it runs. If not, I'm going to be really out of luck here. So watch closely. Did you see that? It went by kind of quickly. Okay, watch again. You sort of have to use one eye to look at the top row and the other eye to look at the bottom row. And if you're strabismic and your eyes don't work together, you're in better shape. Okay, so watch one more time. So, on the top, some response that was significant was detected over a short period of minutes when the person was smoking and that same person did not have any activation, apparently, in the same method when they were at rest. So that's encouraging, right? But now, okay, that's not really what we want to do. So we go to applying this. Okay. Whoops. To men and women, take my word for it, it's a woman on the top and a man on the bottom. And I think the bottom movie ran already. It'll come again. And then the woman, and there's a difference, which we noticed, which started to really interest us. Let's see, the movie, I'm trying to get the movie on the bottom to run first, which is the male. Okay. Is anything moving? Now, on the bottom, there it is. Okay, so the male, well, on my screen, there's right ventral striatal activity, and in the woman, is it running now? It did run? Okay, there's more dorsal activity. Now, of course, this is just one woman and one man, which, of course, were picked because they're perfectly aligned with our total results, which I'm going to show you now. Okay? So, and that's this. So in fact, we would eat popcorn in lab meeting, we would watch these movies over and over and over, and we had convinced ourselves we could actually tell the difference between men and women. So if I'm in America, I say that you all should be happy that your tax dollars went to a study that can show that men are different from women. Luckily, you're mostly Australian and you didn't actually contribute to this study. But in any case, we can quantify the results. And this was what we got in the first study, which showed that the spatial extent, the probability of activating voxels in the ventral striatum, and particularly in the right ventral striatum, was different. It was greater in men than in women. And the number, the spatial extent of activation is that black bar, and the women almost didn't activate their right ventral striatum. That is, they didn't have any detectable dopamine response during smoking in these studies. Okay? And there you go. Right ventral striatum was significant. I'm not sure it's unilateral. It may just not be may not have been enough people. We could, as confirmation or support, we took a different approach, which was not to look at spatial extent, but to think in SPM terms and say, okay, remember I told you that those curves are parameterized functions, and if we compare the magnitude, the height of the dopamine curve at each voxel and ask where is the magnitude of the response higher in men than women, you get essentially the same area in right ventral striatum. So that was supportive. Both spatial extent of activation and magnitude of activation were in support of this idea of a sex difference. Okay, now you ask, well, why are you even interested in men and women and sex differences and smoking? And it turns out that men and women smoke for different reasons. Men are more sensitive to the nicotine effects. They're more reinforced by nicotine, and that implicates reward and ventral striatum. Whereas women report that they smoke for social reasons and that they're sensitive to cues and they smoke out of habit. They're not sensitive to the nicotine level per se in the cigarette, and that implicates dorsal striatum. So this was all starting to become an interesting story, to which the IBIS asks, okay, but if not ventral striatum, something must drive the women to smoke, right? We did match them for dependence. And so this I'm not going to go into too much, except to say that we asked the question, well, maybe it's the speed of response to the cigarette that is important for encoding reward. And so therefore, in an SPM sense, we asked, where in the dorsal striatum is there a locus that the women respond faster to the men? This is sort of a potential area of importance, and we could find such an area, sort of preliminarily, that where the women, their dopamine curves took off faster. So they were responding faster. Maybe that's important. But in any case, we have this method. We think it's interesting and promising, and it was enough to sort of suggest that we could use it in the following way: to identify a signature of addiction, a signature which is both spatial and temporal, and then to ask the question, does it get altered by treatment? Okay, now there's a little American joke there. Anybody recognize it? Okay, that in America, John Hancock, who was the president of the Continental Congress, his name was biggest on the Declaration of Independence? Yes. Thank you. And so when you say, give me your John Hancock in the United States, it means give me a signature. Okay. All right, so finally, these are preliminary results of just exactly that design of a medication study, looking for differences in medication. How am I doing? A few minutes, right? Okay, enough, that's all I need. Because you guys were answering so many questions, actually. All right, so here's our design. Treatment versus placebo. The treatment is the nicotine patch. It's both men and women. It's randomized, it's double-blind, and it's crossover. And the hypothesis is that given the differences in why women smoke, and I should have said this, that men are 1.5 times as likely to respond positively to the nicotine patch, we hypothesized that there would be some difference in these movies on and off the nicotine patch between men and women. That is to say, there would be a differential effect of nicotine replacement in men smoking versus women smoking. And again, this is the same design, but each person comes back. They smoke both times in the scanner, once they're on placebo patch and once they're on nicotine patch. All right? This is the model again. It just says I'm going to show you data from 8 women and 10 men, and that we're going to compare the magnitude of that dopamine response. First I asked this question, well, the placebo condition should look essentially, we hoped, like the first study, which was no treatment at all. And so far, I'm not sure it looks exactly the same. It's right dorsal putamen instead of right ventral striatum, you know, a couple years of, and 40 more people, and maybe it'll all work out. So I'm not sure we've perfectly replicated. I'm not sure the placebo condition is the same as the first study. But here's the most interesting thing. We're asking the question, and this is preliminary data, are there areas of nicotine's effect on the response to smoking that differ between men and women? That was our hypothesis, because the nicotine patch works better in men than women. And so this is just SPM results asking, it's a little complicated to ask it in the SPM sense, but effectively it's, are there places, this is just men, is there somewhere where the response under placebo is greater than the response under nicotine patch? And we regard that as a place in the brain, a response that is extinguished by the nicotine patch. And in fact, there is a small cluster if you lower your threshold enough so that you have something to show at WFNMB. But what's beautiful about this is that if you ask the same question throughout the striatum, which is what we're doing with women, is there a place in the brain, in the striatum, where the effect of the nicotine patch condition extinguishes the activation that we detect with this method? In women, the answer is 0. So that's in alignment with our hypothesis. And finally, in my last, I think these take 20 seconds to run, this is a man, which of course was picked to agree pretty well. It's left ventral striatum, but remember, I hedged my bets and said that it might be bilateral. And in the, can I run this? Is it running? Did it run? One person nodded out of all of you, so I'm going to take that as a yes. It did run, and that there's another person, also a male, because that's, again, is it running? Yeah, okay, great. So there is something in placebo. It's extinguished by the nicotine patch. All right? And so I would just say this in closing, that there's lots more smoking to do. Thank you for your attention. Okay. Sure.
Okay. Are there any questions for Evan? We might just have time for just one question. Michael.
Evan, is there anything known about the gender differences in expression of nicotinic receptors or their subunits that could explain what you're seeing, the differences in that?
It's a good question. I think there is, this is work by my colleague Kelly Cosgrove, but she didn't prep me enough to answer your question. So I believe there are sex differences in nicotinic receptor expression, but remember, this is dopamine, of course, and so it's downstream.
Yeah, I was just wondering whether those differences correlate with
Could. Okay, so that's a very good question. I don't know the answer, but I should.
Okay.
That was only half a question. Give somebody else a chance.
Yeah, one more question.
Anybody? Okay.
Plus, he didn't know the answer anyway. So.
Yeah, that's right. Give me another chance.
All right. I think everybody's really keen to.
One question.
Oh, we've got a question.
Hey, very nice. But I was just wondering about there's evidence, some evidence that anticipation of smoking and anticipation of a pleasurable activity release dopamine. So
Yeah.
is your timing interval
Wide enough.
yeah, does it
Yeah.
go early enough to deal with that anticipation? And then do you have some instances where you would maybe they think they're going to smoke, but they don't smoke, to see how much of the effect is anticipation as opposed to
Yes.
the actual nicotine delivery?
Yeah, it's a great question. So it's very important that our timing window of where we allow a response to happen starts 5 minutes before the cigarette is given. And the 5 minutes was chosen because that's when the graduate student walks into the room, holds out the little cup to catch the ashes and says, "Mr. Jones, we're about to smoke like we practiced." But it's very interesting to see what Steve was showing with rats and the whole idea of seeing if we can detect dopamine changes with negative reward prediction error, which is really what we want to be doing.
Was there a reason you don't start earlier? Could you, I mean, I guess you're requiring earlier, but.
Well, so the trick is it's a balance. You need enough of your data apportioned to sort of lock down the tracer parameters and then allow, if you allow a dopamine curve to happen anywhere, it's much more difficult to identify or impossible the tracer kinetic parameters. So you sort of have to use part of the study for one purpose and the other for the other. The alternative, but it's experimentally more difficult, is that you could do a whole baseline study on people with no smoking, and then you would have more flexibility to move your perturbation around in time in the second scan, but then you lose people that don't come back and so forth. Okay.
All right, I'm sure everybody's keen to get to lunch, so let's just thank all the speakers once more and close the session. Thanks, everybody.